Thanks for inviting me to give this talk today, and thank you everyone who has logged in to watch this. I actually can't see the participant list, but I assume that it is, vast and full of old friends that I've met over the years. Feel free to interrupt me with questions as we go along. It does not bother me. If I don't respond, it means that I can't actually hear you, so please make Aura aware that you want to ask a question, and he can relay it, because I can see him and Chris. So, all of my degrees are in bioengineering, but I actually also studied history in undergrad, and I find history to be a very useful guide for kind of framing where we go with our science and to understand what role our science can play in the contexts of our current time. So for anyone who's interested in kind of a very brief overview of the history of the science of the heart, it wasn't in the Western world, at least in Western Europe, until the 1600s that our understanding of the closed loop circulatory system really became pronounced. And this was performed or published by Sir William Harvey, a very proper Englishman, who came to this conclusion based on experiments both by dissecting cadavers of animals and by performing what you see here, which are tourniquet experiments and looking at essentially the inflation and deflation of veins. And this understanding of a closed loop circulatory system and the role of the heart in pushing blood against a pressure gradient in that system has really dominated our understanding for hundreds of years. And a lot of our understanding of the heart changed last century with the advent of medical imaging. And so this is one of the original Röntgen scans or x-rays. It's titled Hand mit Ringen. It's actually his wife's hand with the ring on it. And as we all know, what she said when she saw it was, "This will be the death of me." And she was right, because he used her so many times that she developed terrible bone cancer. But medical imaging has become so commonplace that you even see it mentioned in children's books. And so the combination of kind of an understanding of the heart as an integral part of a closed loop system and the ability to start imaging these components kind of collided in the latter half of the 20th century, really with the advent of the age of the automobile. And when you see how the heart is described in literature during that time, what you see is the description of an engine. And this understanding of the heart as an engine with mechanical components that break down over time, that get clogged over time, really drove research into understanding sources of heart failure and potential treatments. And this focused entirely on the study of coronary artery disease as the source of heart failure. And this is not a bad thing. What this graph shows you is the age-adjusted cardiovascular mortality rates due to coronary heart disease, cardiovascular disease, and stroke. And what you can see from the decline, the rather precipitous decline over the last 75 years, in this case actually longer, or the last 70 years, is that there has been a very steep decline in mortality from cardiovascular disease, but it has been driven almost entirely by improvements in treatment of coronary heart disease. And these include the ability to detect the disease earlier, both by the development of diagnostic technology to identify the appropriate individual, using that to understand how to design therapeutic treatments that target the origin of disease, so hypertension, hypercholesteremia, and together these effects help to decrease mortality rates, along with novel treatments like rushing someone to a cath lab. So if you look at kind of the diagnostic imaging that we have today in cardiology, it's really orientated around the detection of coronary heart disease. And so this can be performed either with X-ray angiography and contrast agents, or oftentimes what you'll see now is the use of echocardiography. In this case, this patient has an apical myocardial infarct. And I apologize if the animated GIFs are not rastering correctly on your end. I've had some problems with that on Zoom. But the idea is basically to look for wall motion abnormalities that either indicate latent ischemia, or in the cases of a stress-induced response that might indicate a perfusion deficit in response to stress. There's also been a very large push for nuclear medicine approach, where following injection of a radiotracer, you look for areas of reduced uptake of the radiotracer, indicating reduced perfusion and coronary artery occlusion. And of course, this is actually just a black hole and the first image of a black hole, but it looks so much like a PET scan. So this brings us to kind of a new challenge, which is treatment of coronary heart disease has been very successful, to the point where about half of the patients who are now presenting with heart failure have non-ischemic etiologies. And this is particularly prevalent in what we see in the patient populations in the United States, which tend to skew both towards being more obese, having more comorbidities. So I like to show this graph to frame the severity of the problem and why we do the research that we do in my lab. So if you look at this graph, what you can see is perhaps logically, patients who have coronary artery disease and diabetes will have a very high rate of annualized cardiac mortality. But sometimes it's also very useful to look at where the p-values are not significant. Patients without coronary artery disease, but with diabetes and reduced coronary flow reserve indicative of microvascular dysfunction, have as high of a mortality rate as those patients with coronary artery disease. And this highlights a complex problem because imaging perfusion reserve requires interventions, requires contrast agents, it's time consuming, and it tells you about one parameter, which while it may be predictive in some sense, doesn't tell you about the underlying physiology in particular. And if we look at in non-ischemic heart disease, in particular, this kind of growing field and consensus around the idea of heart failure with preserved ejection fraction, what we find is that our standard diagnostic methods are not particularly useful because almost all of our regimens have been built around imaging of ejection fraction. And the physiology behind these changes is particularly interesting because there's a common set of sequelae. Most interestingly, what we tend to see is the development of interstitial fibrosis, so extracellular matrix expansion, mitochondrial dysfunction and energetic imbalance, a response of cardiomyocyte hypertrophy, and to some extent, membrane disruption. And these are the common physiological things that we would like to be able to measure, but we really can only measure reliably at this point with biopsy. So if we look at the divide where we stand, what we have is a really strong ability to examine a diagnostic window when changes integrate at the whole organ level. But the underlying physiological changes, which are both the early onset changes in these disease processes and ultimately the therapeutic targets of emerging drugs and other treatments, do not have a very strong mechanism by which to identify them. And so what we ask is how can we change our acquisition methods to be sensitive to these changes? So we use MR not because I have a long history in MR or because it's pretty or because the magnets are huge and expensive, but we use MR because it's a very elegant platform for further innovation. So MR is already an FDA approved modality. It's already the standard of care for imaging the heart. It has a huge install base in the United States, Canada, and Western Europe. There's no ionizing radiation, so you can make repeated measurements, but it's a fundamentally underutilized platform for molecular imaging. So almost all the innovation in MR has, and in the heart in particular, has gone into imaging, getting better images of the heart with better contrast between blood and myocardium, and better contrast between areas that take up gadolinium-based contrast agents and those that don't. And this makes sense because if you look in the heart, in every cell in our body, we have 55 molar water concentration. So we have this huge signal. And what we're taught in our MR physics classes is that other stuff from organic chemistry exists, but we're just going to pretend like it's not there. And the reason is logical. The concentrations tend to be low, but these low concentration targets are actually what make up all of the interesting physiology. And so the question that we have is how can we exploit the presence of all the interesting physiology in a way that is more sensitive and more robust than spectroscopy? And the answer is through a technique called chemical exchange saturation transfer, or CEST. And I assume most people at MGH are relatively familiar with this because Chris does a lot of work, Chris and Or do a lot of work on CEST. But for those who aren't familiar with it, the basic idea is when we're imaging with MR, we are imaging this bulk water pool that has very high concentration. When you perform spectroscopy, if you want to target, say, an amine proton, shown here, that has a distinct resonant frequency relative to water, spectroscopy is only going to give you the small blip. And there you are innately concentration dependent, and oftentimes the SNR is too low for measurement. But in CEST, what you do is you apply radio frequency energy at that offset frequency to saturate it with the magnetic moment of that proton. And that saturated proton undergoes exchange with the surrounding bulk water. Now, this process occurs multiple times. And so instead of just looking for a tiny blip, you're effectively labeling multiple water protons with saturated magnetic moment. And this leads to, at the end of the saturation period, a significant decrease in the steady state magnetic moment, longitudinal magnetic moment of water. And in effect, what this means is that we are using the bulk water pool that surrounds all of the interesting stuff that we're targeting as a gigantic endogenous amplifier for these tiny molecular signals. And so after you acquire a data set, you end up with data that looks like this. It's called a Z spectrum. And so the 0 on this axis represents the resonant frequency of water. And so if we saturate at the resonant frequency of water, we lose all of our signal. If we saturate at the resonant frequency of our target, then we will lose water signal as a function of the concentration and the exchange rate. If we apply RF energy at the conjugate offset frequency, we shouldn't see anything because there are no exchangeable protons there. So a really quick visual example of this divergence between these 2 points can be seen in this example shown here. So this is a standard MR image of a phantom containing water, and then 3 smaller sub-phantoms that contain, 2 of which contain CEST-based contrast agents and 1 of which contains saline. With standard MR imaging, you're just looking at the concentration of water in each sample. With CEST, you can say, show me just the contrast generated by the europium contrast agent, show me just, that's the one labeled E, show me just the contrast generated by the ytterbium contrast agent. And because each of these is in a separate acquisition and the contrast is selectively turned on, we can assign each a fictitious color code in MATLAB and then multiplex all of the molecular information, and more importantly, directly superimpose the molecular contrast to the point of origin. So there is no need for co-registration techniques, etc. You can directly say that this contrast value came from this voxel and that corresponds to this voxel on an anatomic image, or an image that's weighted based on anatomy. Now, when we look at this in vivo, it becomes a lot more complex, but also a lot more interesting in a problem-solving kind of way. So if we were only made of water, right, and we acquired a full Z spectrum of a tissue in our body, in this case, we're just looking at the heart, then we should look like this black curve. This is the data that we should give, that we should be able to obtain in a sample of pure water at a field strength of 3 Tesla with the saturation parameters that we use to acquire the data. However, this is what we as human beings look like in the heart, as healthy human beings, right? And the reason why this data diverges is because of all of the underlying interesting physiological targets. So if we look at what makes up our tissues, and we classify it first as extracellular or cellular, then the change in the extracellular volume of water and its interaction with the ECM endows the Z spectra with this offset from a steady state value called, and this is called magnetization transfer. It's a broad offset. If we then look inside of our cells, the creatine kinase pathway and the metabolic components of that, both creatine and phosphocreatine combined, create a significant offset centered around 2 ppm. Similarly, what we have is almost the entire intracellular pool of proteins. So this is both soluble protein and cytoskeletal proteins. And these generate a significant contrast effect at 3.6 ppm called amide proton transfer. And then what's emerging is that we also have phospholipid membrane contributions in the sarcolemma of cardiomyocytes and other cells within the heart that contribute signal on the opposite side of the spectrum. And this is called the nuclear Overhauser effect. And if we look, not only are these the most interesting targets to look at physiologically, but combined, they can explain almost all of the divergence that we see from a theoretical water curve. And what this means is that the acquisition of CEST-weighted data, or a complete CEST data set, can allow us to quantitatively explore all of these sequelae of non-ischemic heart disease across etiologies. So the development strategy that we have in my group is to go in a very stepwise fashion, because we would rather do each thing individually well and then integrate it than rush through the process and do a whole bunch of things less well. So we start by looking at each target individually, optimizing the development of the MR methodology, the pulse sequence development around that. Then we identify relevant preclinical models, and that includes both small animal models and preclinical as in ourselves, because we always test everything on ourselves before we try to get it into people. And then eventually we start recruiting relevant patient populations. This was true pre-COVID, and it's going to be interesting to see how we're able to do this post-COVID. But up until March, we had a very active recruitment program for patients that would most benefit from the translation of these methods. And then in parallel, what we find is that oftentimes our data reveals novel targets that may be what I call CESTable, so things that can be identified by CEST, novel ideas for quantitative development for characterization of patterns that we derive from these images, and further mechanistic studies that have nothing to do with imaging, which I'm happy to talk about offline. But actually 50% of my lab at this point is operating within the wet lab space and has nothing to do with imaging. So this slide, to quote my friend Asaf Gilad, I love this slide and I'm also saddened by this slide because it represents like 7 to 8 years of work and it's all reduced down to a single slide. But the idea, the main challenge of doing CEST in the heart is that the heart is a kinetic organ. It moves periodically. It's centered next to areas of susceptibility. And if you're particularly looking at preclinical models of the heart, a human heart will beat, 1 time per second, between 1 to 2 hertz. A mouse's heart will beat 600 beats per minute, up to 800 beats per minute. And so everything becomes a lot more challenging. And so the question is, how do you take the theory of CEST and then deal with the realities of MR? And rather than kind of go through a derivation of excitation, relaxation processes and encoding, which I do in my MR class every spring that I teach, I will just say that a lot of work went into developing methods so that the CEST effect is encoded into the steady state longitudinal magnetization of the myocardium so that the signal that we measure from that tells us specifically about the CEST effect. And more details can be found in all the papers that are cited on this slide. So the first target that we were really interested in looking at was looking at extracellular matrix remodeling or tissue fibrosis using the CEST effect. And part of this was motivated because I started getting into this kind of work right around the time when nephrogenic systemic fibrosis became a real concern. And there was a big push to find ways to replace the use of gadolinium for diagnostic imaging. And so this isn't so much a CEST effect as the MT effect, which is what we see in the orange line on this graph. And the basic idea is that if you look at the macromolecules that make up the extracellular matrix, the tumbling rates are very high in those protons. And so they have a very wide bandwidth across which they can be saturated. And they saturate very easily. And so when they saturate, they exchange that saturated magnetic moment with surrounding spins in the extracellular water pool. And this leads to a reduction in the steady state signal of water that you see here at either end of the spectrum. When you have remodeling of the extracellular matrix, paradoxically, even though you have more extracellular matrix components, so more collagen, more fibronectin, et cetera, that added torque on the extracellular matrix increases the extracellular volume of water and actually reduces the labeling efficiency. So this MT effect should actually decrease with higher fibrosis. And that's actually also the mechanism, a parallel mechanism by how gadolinium works, because you have larger pools of water for T1 shortening. So there's a clear need for this in patients who can't tolerate gadolinium or need repeated follow-up scans to look at dynamic changes in extracellular matrix deposition. So we started by looking at a mouse model of myocardial infarction. And in this case, we just surgically induced an MI. And we observed a change in MT in scar tissue compared to normal tissue. And this largely confirmed findings that were observed ex vivo by Bob Balaban around the year 2000. We then moved this into a human system on a 1.5T scanner. So in this case, we just used the endogenous MT weighting in balanced SSFP imaging. And what we observed was that in a patient population of somewhere around 50 patients who were being referred for a late gadolinium enhancement imaging as part of their standard of care, we were able to acquire pre-contrast images using basically a dual flip angle approach for balanced SSFP to create images with high and low MT weighting. And we observed very strong correlation between the contrast generated by the loss of MT and the contrast later generated following gadolinium accumulation and inversion recovery imaging. And what you can see in this slide is a comparison of both acute MI in the top panel and chronic MI in the bottom panel. And you see similar areas of enhancement. These maps were kind of inverted, so higher values actually means low MT on our CEST MR images. And this was done to emulate LGE as much as possible. This is an example that I actually, of this work, this is my favorite example. And this is because these are two patients with non-ischemic dilated cardiomyopathy. The patient on the top has no pattern of LGE enhancement. The patient on the bottom has reactive fibrosis in the mid-wall of the septum, shown between the two red arrows. Our endogenous contrast approach shown on the right was able to replicate these two enhancement patterns and could represent a way to differentially diagnose patients with the same disease in terms of risk factor for fatal arrhythmias. So in the study, we also looked at measuring gadolinium partition coefficient. So that was done with native T1 mapping and then T1 mapping post-gadolinium. And we observed that for the identification of focal fibrosis, we had a higher area under the curve value than native T1 mapping or native T2 mapping. And given a lot of the kind of recent data that's come out, that should not be particularly surprising. What's more interesting that we did was the kind of negative control experiment, which you tend not to see in this literature space. And the reason why this is interesting is we didn't set out to do a negative control experiment. So originally we recruited 30 diabetic patients because at the time we'd read that diabetic patients were supposed to have variable amounts of diffuse fibrosis. And the idea was to probe that dynamic range at the lower end of ECV. What we found out was that all 30 of our patients had normal ECV values. So we managed to recruit 30 people without a single person having indicators of diffuse fibrosis. But what we found was really kind of interesting in this sense. Our measurements of MT contrast stayed remarkably consistent across patients, regardless of structure of the myocardium in the left ventricle. Whereas what we observed was that native T1 times did not. And if you look, what we observed was that patients with hypertrophy demonstrate higher native T1 times, likely because of increased intracellular water content in hypertrophied cardiomyocytes, whereas our MT values did not show any consistent trend across mass to volume ratio.
Moriel, may I stop you for a quick question?
Sure.
So I think Chris Wynn asked if for acute MI, there shouldn't be any fibrosis formation yet. So what is the mechanism behind the CEST signal change?
So the answer to that is it's just you have a lot more water present in the edema, right? So this was, you have a profound loss of MT and a lengthening of T1 and T2 times. So in balanced SSFP, you get elevation of the signal because of that. However, if you reference in that paper, the paper that was referenced there in the supplement, we actually showed how this is not strictly a T1 or T2 effect, that T1 changes in T1 and T2 times just based on the closed form equation for balanced SSFP could only explain about 20% of the signal change, and that the rest comes from loss of MT, just because you're obliterating the organization of the ECM in that area.
Thank you.
So one of the first patient populations that we were interested in applying this to were patients on dialysis. So we know that patients on dialysis have high mortality rates from fatal arrhythmias linked to the development of fibrosis. It's actually about 20% per year after initiation of dialysis. Existing heart failure drugs at the time were largely ineffective, although there's kind of a new class of soluble guanylyl cyclase inhibitors that seem to be relatively promising. The only methods for looking at fibrosis were invasive at the time, which were biopsy, and which is highly contraindicated, and this represented a fundamental barrier to the development of new techniques. And just to give you guys an idea of how bad dialysis clinics or the dialysis epidemic is becoming, when I was visiting Walt Whitchie at Penn, I went and walked around a little bit to visit Liberty Hall. This is where the American Republic was founded, should it last through the next week. And the building across from it over here, the white building with the beautiful columns, used to be a mercantile and exchange building and is now a Fresenius dialysis clinic with about 200 beds. And if you read about community action in the East Bay, where I live, you hear a lot of discussion about how in lower income communities, it used to be that every kind of corner building was, a store that sold junk food and liquor, and now they're all dialysis clinics. So this is a very clear need in the population of patients in the United States, and is only going to grow as the obesity epidemic becomes a kidney failure epidemic. So before the discovery of nephrogenic systemic fibrosis, studies performed at the University of Virginia showed that in 86% of patients who are on dialysis, one of three prevailing patterns of fibrosis were observed. The first, in about 10%, was thin scar formation relative to a prior infarct. In about 80% of the population, it was diffuse subendothelial enhancement. And in another, about slightly less than 10%, you saw focal fibrosis at an RV insertion point. And a decade later, we went back and looked over all of our data, and we were able to observe similar patterns of enhancement in our patient population to what was seen before. These are not the same patients as the top row, it's just similar patterns. And in our patient population, we were able to bring back a handful of them for follow-up imaging 1 year after the initial imaging time point. And what you see here is the 42-year-old female patient who'd been on dialysis for 2 years, You can see that she has pleural effusion outside of the myocardium, but the values for the contrast metric inside of the myocardium were all relatively low. A year later, she returned wearing a heart monitor, an arrhythmia monitor, for what she described as her heart palpitations. And you can see that the increase in contrast observed, particularly in the septum, is characteristic of reactive fibrosis that you see in that area. She had not had a prior MI. We spent some time at the time looking at how we could describe this. So we looked at the development of cumulative distribution functions for normal controls versus patients, and showing the data in the form of a bullseye plot. And this would allow us to describe quantitatively, both regionally, where areas had become much worse in terms of density of fibrosis, and then providing a global metric for the increase in global fibrosis. It's an intrinsically hard problem because you have a diffuse disease spread over a hypertrophied organ. And so finding a way to normalize for size of the organ, to actually look at the density of a diffuse process that doesn't just average the values out, was a little bit tricky, but that's what we did in this case. Another kind of project that we built off of this was trying to describe the morphology of these patterns, which are kind of intrinsically scattered and diffuse. And so a master's student that I had at the time, Zainab, created a system where we could basically from each part of the heart identify the RV insertion point, convert the structure of the heart to a set of polar coordinates to essentially unwind the heart into a linear space that was then created in a matrix that ensured the physical connectivity. So every voxel in this, or every square in this matrix actually touched the voxel that it was connected to spatially, as you would expect from the orientation of the heart. And then once this was in a space of this format, we were able to perform fractal analysis, because if you look at the kind of original studies of developments of fibrosis in tissue, what you see is that the fibrotic tissue actually expands outward in a fractal pattern, consolidates around the base, and then again expands outward in a fractal pattern. And in case you're wondering, yes, I have 2 small children. Yes, my 3-year-old was obsessed with Frozen at the time, and that is actually how I thought of using fractals to analyze this data. So when we looked at this, Zainab implemented a box counting algorithm that allows you to basically characterize the space filling morphology of given voxels that were above a threshold value that we knew to correlate well with voxels that would enhance with late gadolinium enhancement. And we're able to see a very interesting morphological transition, which is that as the scar tissue increases in the volume of the heart that it occupies, it morphs from being planar in 2 dimensions to being fractal in 3 dimensions, and then eventually entirely filling the 3-dimensional space. And the interesting thing is that in the intervening years, since we first started showing this data, a number of groups have actually started looking at fractal characteristics of the heart in more detail. So one of the big kind of endpoints that we were interested in for this study was looking at a new way of how to characterize fibrosis in dialysis patients. So if you look in dialysis, in the literature of dialysis patients and what's called cardiorenal syndrome, about 20 years ago, some people noticed a correlation that existed between patients who had fibrosis and patients who were hypertrophic. And that created the moniker that hypertrophy is just a surrogate marker of fibrosis, and that has been used ever since. And our findings very clearly showed that hypertrophy is not a good surrogate marker for extracellular matrix remodeling in this patient population. All of these patients that you see in this example are hypertrophic, but as you can see, they have very different enhancement patterns. And in that paper, you can see a more robust analysis of how it's kind of a situation where, you know, a square is a rectangle, but a rectangle isn't a square in terms of the relationship between hypertrophy and fibrosis. More importantly, if you looked at the literature at the time, a lot of the drug targets that were being examined or the blood biomarkers that were being purported to represent fibrosis in these patients' hearts had very strong correlations with hypertrophy, but very loose correlations with our measure of fibrosis. And in fact, what we found was that other markers that people weren't looking at correlated very strongly with the level of ECM remodeling. And this finding has actually been what's basically spurred a totally new direction in the lab. So my graduate student, AJ, is shown here. He's actually a very serious guy. He's not always so goofy. And I recruited a whole army of undergraduates, half of whom at this point are either Knight Hennessy scholars at Stanford, medical students at UCSF, graduate students in engineering programs, or still in the lab as an undergrad. to create an entire kind of biomechanistic examination of where the source of these particular markers comes from and how to treat them. And this is something I'm happy to talk about separately in a one-on-one talk. So as we continue developing methods to look at fibrosis, we're also interested in looking at metabolism. So it's long been known that there's a need for a high sensitivity method for assessing metabolic remodeling in the failing heart. A lot of work has gone into P31 spectroscopy of the heart and looking at the ratio between phosphocreatine and the kind of subunits of ATP, and the reduction in this, a reduction in this ratio is indicative of failing heart. And a lot of this corresponds to the breakdown of the creatine kinase pathway. And that results in, and that is caused by the inability of mitochondria to adequately produce ATP to match demand. And that results in a downregulation of the levels of both creatine and subsequently phosphocreatine. Now, the amine protons that are found in creatine resonate at around 1.8 ppm, and the amine proton in phosphocreatine have resonances at 2 and 2.5 ppm. And the impact of this combined concentration that is around 50 to 60 millimolar in cardiomyocytes leads to a strong deflection around 2 ppm. And so in kind of a first proof of concept study, we looked at the correlation between this contrast and just the underlying concentrations in a phantom. But what we were really interested in looking at was mouse models that were relevant for humans. So this was at the start of my assistant professorship, and by coincidence, my wife left for Thanksgiving to visit her family, and I didn't really want to go with her. And so I stuck around, and I was tired of writing grants, and I really missed just doing science. And so the lab next to me was looking at metabolic effects in mice fed a high-fat diet. In case you're wondering, the high-fat diet mouse on the left was fed what Americans eat, and the low-fat diet mouse was basically fed what French people eat. And the interesting thing about this mouse model is that it takes about 42 weeks to develop systemic changes in the left ventricle. And what you can see is, in response to a beta-adrenergic stress test, you can see changes in contractile function at about 28 weeks, and you can see changes in perfusion reserve at about 18 weeks. And the time point for this happened to just align with 14 weeks. And really, I was only doing this because I wanted to test the sequence that I wrote. And I didn't even really pay attention to what was going on, which mouse I was scanning. And after the fact, I noticed that I had two pretty clear groups. And so I went back and looked at what we were seeing in terms of classifying the mice correctly. And what we observed was that in the obese mice at 14 weeks, we saw a profound reduction in contrast generated by metabolic markers, creatine and phosphocreatine. And this was despite preserved fractional shortening, as well as other markers of systolic function and diastolic function in these mice. We then tried to repeat this experiment in human subjects. So we recruited healthy and obese human subjects. And not surprisingly, we kind of specifically looked for this. We looked for adults who had normal ejection fraction, normal left ventricular mass index. We looked at characteristics of contractile function. So we used myocardial tagging to characterize strain. We looked at global longitudinal shortening. We looked at all the parameters that you could actually assess from imaging of contractile function, and they were all normal in this population of adults. However, what we observed was high variability in the subsequent contrast from metabolic markers. And the one thing that this correlated with, it didn't correlate with any measure of contractile function, but it did correlate very strongly with the level of visceral fat, essentially the density of visceral fat. And that speaks to kind of an emerging hypothesis of the role of infiltration of lipid droplets into cardiomyocytes, leading to mitochondrial damage and reduced energetics. However, more recently, we've been interested in the idea of how can we acquire all this data simultaneously. So everything I've shown you up until now is looking at one target in isolation. And what we're really interested in is looking at all of these parameters in a single scan so we can perform integrative physiological imaging. And one of the real challenges when you're performing this at 3 Tesla is dealing with inhomogeneities, particularly B1 inhomogeneity. So this is actually data that is acquired from one of my graduate students who is a healthy young adult. And what you can see is in the septum where we roughly get the power, the saturation power that we prescribe, that's been optimized to generate all of these contrasts. You can see that we get a very nice Z spectrum with all of the underlying markers that we're interested in measuring. However, as you start to rotate around the heart and you have inhomogeneity in the B1 field, higher powers will lead to different appearances of the Z spectrum that ultimately may prevent you from accurately measuring the underlying markers. And in this case, may make a healthy patient look like they have no metabolic function whatsoever. And so one of the real challenges is creating as uniform of a B1 field as possible so that you know that the CEST contrast that you're getting reflects the underlying metabolites and not the input parameters that you used to generate that CEST contrast. So one of the ways that we're doing this is through collaboration with Will Grissom and Vanderbilt. So we measure a B1 map so that we can actually characterize the deposition of power spatially. And then we use a technique called B1 tailoring that reads in that B1 map and then creates a spatial, essentially a spatial spectral RF pulse that is optimized for that individual. And that allows us to create as uniform as possible of a B1 field for generating contrast. And I'll show a slide on the next slide, the impact that this has. But what this allows us to do is both acquire B1 optimized D spectra, and we therefore know what the actual B1 was at a given region in the heart. And that allows us to perform essentially a library lookup approach, or some people might call this a fingerprinting approach, although it's quite different from MR fingerprinting. And it allows us to then convert the contrast measurements that we get for each metabolite from CEST contrast, which is a rather arbitrary number, to then matching it to a library that allows us to report that in units that are relevant. And that means units of concentration. And it allows us to output our parameters in terms of concentration of metabolites, intracellular protein fraction, collagen volume fraction, et cetera, and report how confident we are in that fitting. And so a lot of this depends on the development of this optimized B1 tailoring, but I just want to show you an example of why this is so crucial. So with a normal Gaussian saturation pulse scheme that is used, for example, to look at creatine, you will get a spread in B1 values across the heart. And that results in a pretty significant shift in the saturation parameters. And this means that you'll get a big spread in the subsequent contrast that is derived from that. Whereas using the tailored pulse, you get a far more uniform distribution of values. And when you look at the subsequent contrasts that are derived, it's easy to see a very big spread and, in fact, values that would be consistent with unhealthy tissue when you don't optimize for B1. Whereas the optimization for B1 allows us to obtain consistently healthy values. So we're running a little bit short on time, but I want to touch on some of the kind of new directions that the lab is going that are things that may eventually actually work their way into human studies. So lately, I've been getting very involved in the NIH's somatic cell gene editing consortium. And it's a very high growth space because there's a lot of need. So if you look at in vivo gene editing, I'm sure half of you guys on this call are like, yeah, of course, you're at Berkeley, CRISPR, CRISPR, CRISPR. So yes, I have joined the CRISPR train. But if you look at in vivo gene editing, there are so many black boxes that exist that relate to actual assessment of gene therapy. So a lot of work is going into the development of new CRISPR, new forms of CRISPR, new guide proteins, better ways to edit cells. And a lot of work is also going into the development of adeno-associated viral vectors or other vectors for the organ-specific delivery of that gene editing machinery. But when it comes to a trial on a patient, all that you know is what you administer intravenously and if there is some ultimate functional outcome. And all of the stages in the biological transduction cascade that matter and where these therapies almost overwhelmingly fail remain completely opaque without some kind of after the fact biopsy. So the stage of endosomal transport of a virus, endosomal escape, subsequent integration of the transgene and transgene expression are all left unmeasured. And this is a real opportunity area for MR. Because if you can repeatedly probe these measurements non-invasively, then you can actually characterize where a therapy failed, whether a patient would need a top-up dose, and whether expression of the transgene A was effective or B led to maladaptive responses in terms of the physiology of the target cell. So for targeting, looking at transgene expression, Chris and I have both worked with Asaf Gilad and utilized a genetically encoded CEST MR reporter gene called the lysine-rich protein. And this was really more of a proof of concept study on our part. So the lysine-rich protein has a ton of lysine residues that have a lot of amide protons that resonate at about 3.76 ppm offset from water. And what we did was we cloned this into an AAV9 vector. and then used it to probe gene transfer in the heart. So in this set of images, what you can see, this is following direct intramyocardial injection of an AAV9 vector encoding for LRP. And you can see that in the area where the red arrows point to, when you apply RF energy to turn on the contrast from LRP, you see a loss of signal intensity in that region of the heart. And this leads to CEST contrast that is most focal around the inferior RV insertion point with a little bit of contrast in the septum. And this corresponded spatially very nicely to what we saw when we did immunostaining for the V5 marker that accompanied the lysine-rich protein. The advantage of this is not that you can just replicate histology or in vivo bioluminescence, but with MR, you can go back to the exact same point in tissue over time and look at changes in gene expression patterns when you use a transgene that's CEST active. And what we could see following intramyocardial injection was not just that there was a time course for gene expression, but that for each mouse, we saw a slightly different time course. Some mice saw a significant reduction over time in expression. Other mice saw a delayed transcription response. And as a more human relevant study, when we looked at intravenous injection, so one thing to note, I like to show all my noise in all my images. I don't believe in filtering it out. What you can see is this is a low contrast to noise ratio measurement in this particular study. And so there are, in saline infused mice and empty vector infused mice, there are going to be some voxels that appear noisy. And what I was hoping was that all of our mice that received the lysine-rich protein vector would look like the one on the right, where we would just see like, very vast and strong CEST contrast all through the heart. But the reality is that there was only one that looked like that, and almost all of them looked like these two labeled average LRP. And I was kind of flummoxed by this at the time, but I contacted Ben Greenberg, who is basically ran one of the first cardiac gene therapy trials at UCSD, and I asked him if they had seen anything similar or what to make of this. And he pointed out that our contrast seems to be focused around the coronary territories, and that there's a very steep gradient away from the coronary arteries in terms of the contrast that we measure and presumably transgene expression. And that this is what they saw in a lot of the patients that proceeded to heart failure and explantation, that the ones that didn't respond to upregulation of SERCA2 expression in what was called the CUPID trial, that they tended to have very steep gradients of gene expression away from the coronary arteries, whereas the ones that responded well and then later on died and they performed a histological sectioning seemed to have had some kind of preserved and less steep gradient away from the coronary arteries. Now, this is only part of, measuring transgene expression is interesting, and people at Johns Hopkins are doing a lot of work to come up with new transgenes that are more likely to be approved by the FDA, because lysine-rich protein probably won't ever be approved by the FDA. But this is only half of that black box that we're trying to uncover. One of the things that a real kind of twist of fate when I was working at Berkeley was we have a guy named Dave Schaefer who heads our Quantitative Biology Institute, formerly also ran our Stem Cell Institute, who has spun off a couple companies on engineering novel variants of adeno-associated viruses for organ-specific targeting of gene editing. He's a very rich man these days. And in just talking to him over coffee, I was asking about how could we modify the surfaces of AAV vectors so that we could track them, perhaps conjugating them to some like salicylic acid that has a strong CEST contrast around 9 ppm. And he referenced me to a paper that he wrote a long time ago, which tried to do that in order to evade the immune system. But what they found was that it was easiest to target a lot of lysine residues. So AAV2, for example, has each AAV2 particle has 1,080 lysine residues that face the surface. Conjugating them to something pretty much limits their ability to bind to the target cell. And so we started asking, well, what if we actually just used the surface proteins, the capsid proteins of these viral vectors as contrast agents? Because we were trying to track expression of 50 lysine residues in a row, and here comes a viral vector that has 1,000 all over the surface. So we started just by looking at an NMR scan of a sample of AAV2. And of course, if you don't suppress the water signal, you're not going to see anything. But if you suppress the water signal, what we observed were there were actually noticeable peaks around 3.6 ppm, 3 ppm, and 0.6 ppm. And these correspond to the amide protons and the hydroxyl protons on the surface on the capsid proteins. And so we started to ask, could we actually detect these by CEST? And so if you just acquire a CEST scan of very low concentration polyL lysine that's consistent with background signals in biological tissue, you get a Z spectrum that looks like this, and you get no real CEST contrast across the MTR asymmetry spectrum, the subsequent kind of range of offsets. But if you scan a concentration of AAV2 particles, you observe a very big deflection, interestingly at 0.6 ppm, and a smaller, although still relevant one at about 3.6 ppm, but it's an order of magnitude lower. And what's interesting about this is that it's fairly consistent across radio frequency or saturation powers, and it follows a logical curve based on the exchange rate of what we think that we're targeting. And it has pH sensitivity. And what it means is that as the pH decreases, and I'm just showing some small examples here, what you can see is as the pH becomes more acidic of the solution that it's in, you lose contrast both at the higher peaks, but you gain contrast, you lose contrast at 0.6 ppm and more acidic solution, but you gain contrast at the other offsets in a way that we didn't see before. And what this tells us is that potentially this can be very useful for tracking gene editing, and I'll explain why after the next slide. The other thing that we noticed with this was that this is just an example of AAV2, but we observed a concentration dependence of the signal, and we expect it to be nonlinear because when you add more viruses in a given setting, your labeling efficiency goes up nonlinearly. And the advantage of this is if you look at the transduction cascade, when viruses are trapped inside of an endosome, you have very high labeling efficiency, but it's very acidic, so the exchange rate changes dramatically. When they escape the endosome, then they're in the cytosol. You have a different labeling efficiency, but a cytosolic pH value. And so that completely changes the subsequent signal that you're going to get. And what this means is that you can actually perform exchange rate filtering based imaging and then start looking at the relative ratios of how much of the virus is still trapped within an endosome and how much has successfully escaped. And then beyond that, you can also couple this with the other physiological imaging approaches I spoke about earlier. So you can actually start linking cause and effect. If a virus successfully transduced a cell and transgene expression was successful, what were the physiological outcomes? And this creates a very powerful potential integrative imaging toolkit that's entirely non-invasive and can help to guide the development of further therapies. Just as an example, this isn't limited to viruses. We also performed some studies where we looked at cell labeling in imaging with CEST, and we're able to properly image the survival or rejection and elimination of cells that were labeled with the europium-based contrast agent. And that contrast agent resonates well outside the range of any endogenous markers. So it's yet another target that you can add. So you can start looking at cell survival as a function of underlying myocardial physiology. And then the final thing that I just want to touch on, because Chris mentioned it and I knew he'd wanted to ask about it, is moving outside the heart. So recently we got really interested in looking at renal imaging with CEST MR. And this also harkens back to some of the first studies that were done by Bob Balaban, where they actually looked at just the presence of urea. Urea is, a compound that is found in increasing concentration in the kidney, and one of the main goals is to remove urea from the blood and expel it in the urine. And clearance of urea and other uremic compounds is crucial. And what we see in renal failure is a loss of the urea gradient, and the place where many new drugs and therapies oftentimes fail is in renal toxicity. And so there's a strong need to measure renal toxicity in a way that's more sensitive than creatinine assays, urine creatinine assays. And a non-invasive approach that uses imaging could give you spatial sensitivity and perhaps even stronger sensitivity to underlying components of renal function. And so we were able to show that, yes, you can get CEST contrast with urea as a function of concentration, but looking, in this case, these are all mouse studies, looking in the mouse kidney, we were able to observe, as expected, an increasing urea concentration from the cortex to the inner medulla and papilla, where the concentration of urea hits the high hundreds of millimolar to molar range. And more importantly, we were looking at whether you could actually administer urea to enhance this level of contrast. And in the first paper that Soo-Hyun published last year, I guess this year in MRM, we were able to show very nicely that if you inject urea, you can detect the presence of urea and kind of the rate of urea recycling as a measure of renal function. But more importantly, we stepped back and started looking at how the best way to quantify this is. And what we realized is that in our first study, we were really under-determining the contrast. So what you see here are the same Z spectra from the inner medulla and papilla of a mouse fit with the appropriate number of pools. So when we only use 4-pool fitting, we tend to actually miss many of the metabolic markers and the urea marker. And the importance of fitting with the correct number of pools allows you to more accurately quantify the underlying urea concentration. And what we observed by actually correlating it to kidney homogeny was that we were able to observe a reasonably strong correlation between CEST contrast and underlying urea concentration. And this was meant to be more foreshadowing. Be on the lookout. So here in the second paper is about to be in press at MRM. I guess is in press. It's about to be on early view in MRM. And here he dives really deeply into the importance of T1 correction, the use of standard post-injection times, kind of analogous to late gadolinium enhancement, the application to renal failure models, and perhaps most interestingly, one thing that we found was that if you normalize for the baseline CEST contrast of urea and then administer either urea or saline, the contrast really comes from simply the increase in the volume in the inner medulla and papilla, suggesting that even saline could be used as a sensitive CEST contrast agent to look at urea kinetics in functioning kidneys. So I'd like to conclude here and leave a minute or two for questions or more. I'm happy to stick around longer. But what I hope you took from this talk today is the importance of developing newer imaging techniques that don't just look at one target in isolation or perform a series of different scans to try to kind of piece together different parts of a story across different scales. But the idea of refining the way that we perform imaging so that we're sensitive to multi-scale biology in order to really understand the physiology of the heart or of the kidney or of any other organ in a way that gives us a more robust and rich data set by which to both understand disease processes and be able to understand the efficacy of treatments that target any kind of dysfunction of those processes. And this can provide an important framework eventually for personalized medicine. So there are a lot of people that do the work. I just talk pretty about it and write pretty about it. And the people who really do the work are acknowledged on this slide, as well as the bumper stickers that fund it. And in particular, I really need to thank people from Siemens who have really bought into this and are helping us develop a more streamlined platform for cardiac CEST MR that they hope to soon turn into a whip. And then, of course, the people who really motivate everything shown down here below. So thank you. Hopefully we have time for a couple questions.